Hi, Dr. Crumb. Hope you're doing well and hope your family is doing well and that you guys are happy and healthy, as I keep saying. Um, the questions you gave us were to talk about mitosis and meiosis and respect to the chromosomes and the subtle differences between the two. The start of cell division kind of has interphase, and interphase is, in my opinion, isn't really a phase, but it's still a part of mitosis and meiosis because it's the growth is happening, the DNA replication is happening, the G2 cell growth is happening. The cell is just getting bigger and getting ready to divide. There's no division happening. It's just it's go time. It's time to start buckling up. Prophase is where chromosomes start to condense, as you see. I almost drew all 23 pairs. um, the spindle are starting to form, and the nuclear membrane is starting to break down. I mean, this membrane is getting ready to say no more. Metaphase, I kind of blew up a little bit, and it's the lining of the chromosomes at the equatorial plate. And the spindle fibers are starting to align and attach to the chromosomes and getting ready to split. And they attach on connectachores. I hope I didn't botch that one. I could spell it, but I couldn't say it. Anaphase is really after the splitting and the chromosomes. The homologous chromosomes have been split and have now gone to their respective poles or ends, and they're about to form the new cells or the new, and it starts with telophase of it having more sister chromatids or respective sides, and they're prepping for nuclear invo. It's really the opposite of prophase, as if we going back of the chromatids are condensing and now they're breaking down and going back into their regular DNA strands or ready for replication and casual, cell processes. And then as you see, we're back to new daughter cells of mitosis, and it has a new envelope, and cells are back to that interphase looking, casual, ready for anything, buckle up time. Now, meiosis does not look the same as mitosis. It'd only be easier, right? Ha. So, prophase is a kind of a lot like the prophase of meiosis, and there are a lot of similarities, and the really subtle differences is that there's a little bit more cells to be made. So you got to figure out where the DNA is coming from and where it's all going and how it's equally divided and kind of things like that. And the chromosomes condense in prophase 1, and the nuclear envelope starts to break down, and crossing over occurs. Ding, 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 ding. Man, I should get a marker. Let's see. Let's see what we got here. So this crossing over is a lot like, we are going to mix and match. So if crossing over is these two chromosomes, let's say they look like that and they're that color, and I think this is a different color, and then we'll go, that's a different color, and this is a different color. And this crossing over process is these specific genes of the homologous chromosomes are going to be interchanged with certain sections and necessarily swapped or crossed over to exchange genetic material as they are next to each other. So they would start to look like, if something like this was to cross, this one might look something along the lines of finishing this X with an extra blue over here, and vice versa with this side over here with a little bit of black on that side. Whoa, look at that. That's crazy. And this is adding genetic variation as you are exchanging DNA from chromosome to chromosome. The next step would be metaphase 1, and we've seen metaphase as before. Nothing scary. It's lining up on the plate, but this time it is. Look at that. You have the tetrads. Tetrads are two homologous pairs that are combined, giving it tetra, that 4. So now we understand. We got two homologous pairs and coming together. It's starting to all make a little bit of sense. And this one is also an addition to genetic variation, as the order of where these are located on this equatorial plate is random. So when they start to, as we remember in mitosis, we're getting ready for splitting, and if there's no orientation of where these things are, well, the differences of which type of crossover they had, where they are in this line of equatorial, is going to decide which cell they go into, and it's all this big variation of which one goes to where and which cell later on down the line. Now, anaphase, we have these tetrads that are now split, and now they look like what? These look like homologous chromosomes again. And they're split back to their side. And we go back to telophase. And now this looks almost like we're getting the hang of it. We're back to mitosis again. And this telophase is now the nuclear envelope is starting to develop. But instead of it necessarily condensing back to DNA strands, it's going to prophase 2. And prophase 2 is just like prophase